Hello, students. Very good evening. Welcome today. Our last session, last day of our session of MBBT. It feels nostalgic. We had such a great day, 10 days. We're going through a lot of techniques, a lot of things. But today, finally, we had a streaming of 720. So I'm very happy that it started to work at 720. But I think you didn't have any much problem, I guess. Uh, so things will be fine with you. Sorry, it's happening on the last day because my internet speed got increased, so I can do 720p now. Earlier my internet speed was less, so I couldn't do it. So how are you all, students? How's your day? How's life? Where are you standing up at the moment? All going fine in the current of life? Okay. So today I will be dealing with your last day with the applications of recombinant DNA technology and related to that how they are behaving in the human medicine, plant, agriculture, environment, various sectors. Evolution. I'm not even related to that. So that I'm sorry I couldn't help you much in that thing. Evolutionary genomics. I'm more into the molecular biology and biochemistry stuff. Uh, Evolutionary genomics. To study about genomics at the evolution level. hmm This phylogenetics thing is coming up in this. Okay. Systematic biology is there. Okay, okay. No, I'm new to this field actually. Drowned under work. That's good actually. Busy mind is the best mind. Keep yourself busy in things. Being free it's worst. So being busy, mind is always fresh and it's ready to cope up any stress things. Person don't get ill very easily. That's what I have experienced in Germany. I never had a fever or never had a cough or never had any disease while doing my PhD or masters. Hardly 3 years or in 7 years actually. Of course, the weather is such clean that you do not get across these diseases, but still your inner immunities get so stronger because life is so busy that you diseases doesn't even come nearby to you. So that's what the Western culture it seems to be like. But there's mental pressure always. So, but your publication thingy is you have to do it on your own. So I had shared with you already some free articles. Did I? So just go through these journals. You might get lucky. Guys, send it again on the. Just check these journals. Growing position for MSc in biochemistry position. So biochemistry is highly in demand. If you just check, it has a lot of future perspective as per the in academics field also. So there's nothing like all fields have their own demands and their things and their expectations are different as per the needs. So don't think much, just go with the course and see the faculty also while you're doing your course. Yeah, send it to generals actually. I am here to just check that you have done it nicely, but send it to the generals. So that's where it will get published. So I cannot do anything in publication. MSc in zoology. So that's also very interesting. I could go ahead with that too, where you will be dealing with more of the animalia part, I guess. Uh, cell culture and all these will be dealing in that part. So on that note, let's start today's lecture. And one more thing, if you have not subscribed to the channel, please subscribe it by clicking on it subscribe button here. Subscribe. This will help us to grow vividly. So I have 588 subscribers. So I needed 1000 to start my YouTube channel. Uh, being working at commercial level. How to get into biotech and pharma companies after MSc. So there are a lot of positions, there are a lot of advertisements are still happening from September onwards. Earlier, because of the lockdown and COVID condition, there was not so many opening were there, but now a lot of positions are available, especially in the Bangalore side. There are a lot of chances there to get into Hyderabad also. In the north, it's not so much, but in the south, it's a lot. It's a lot. So there are a lot of positions are coming online. Just make a good connections on the LinkedIn. So LinkedIn is the best place to look for jobs. So people from the HR department, they are posting daily some new positions. So it's the best place. Just get into the company's connections. Just be in touch with the CEOs, with the HR department, with the people inside there, so that you can get it. So to pursue career engineering, which is the best major from the list below? Biological sciences, biological engineering, biochemistry, biotechnology, and genetics. So mainly, if you are pursuing genetic engineering, and the major, to go for the major, it's up to you actually. I will not suggest anything. It's up to your suggestions. Which field you are stronger. You're stronger in biochemistry, then go for biochemistry. You're stronger in biotechnology, go for biotechnology. It depends in which field you are feel stronger. Up to you. Pursue PhD, material science. Which country is best? Material science. Sweden is good. Denmark. You can look for Sweden, Denmark, Germany. There might be you have scholarship. Some might have free universities. So you can study there for free, being an international student. Or you can go for Canada also. Uh, it's all freely available. Oh yes, Switzerland. It might be expensive there to study. To stay there, everything is extremely expensive. So shall we start? So they are not free. Okay, I was told that they are free. How much is the processing fee? Yeah, living costs are high in Switzerland. बच्चे अभी Abdija, how much does it cost to publish your things then? If you're paying like 2000 rupees, it's really good money worth to have your own review at the starting. So might be have nominal fees, not so much. So today it's all about applications related to field of health, agriculture, environment, and industry. How recombinant DNA technology that we have studied all these techniques, all we have studied so far, how they are correlated to these fields. So from your hybridoma technology to cleaning the environment, creating antibodies, or making a tolerant free plant, stress tolerant free plant, and creating some vaccines. So all is possible. Uh, all are possible in this field in the with the help of recombinant DNA technology. So let's concentrate applications of recombinant DNA technology in health. So recombinant DNA technology is a wide spectrum in improving health. It helps in treating defective genes or introduce new ones. Applications, laboratory tests, parental diagnosis of genetic disease, all these comes into the applications. First is production of vaccines. We introduce live attenuated organisms to acquired immunity, and then recombinant DNA technology can be used to clone gene for protective antigens, proteins. So they are being used for the production of hepatitis B vaccine, influenza vaccine, HIV vaccine, mouth and foot disease vaccines already. So production of recombinant hepatitis B vaccine, how it is done. So here you can see that there is hepatitis B vaccine, and we take the DNA, human hepatitis B antigen out of that from the virus, and we insert it into the bacterium plasmid to create a recombinant DNA. And this introduction of recombinant DNA is done into the yeast cell. Then recombinant yeast cells is put in the fermentation tank, where the cells multiply producing HB hepatitis B antigens in fermentation tank, so which will produce further your HB antigens, and from there you can create, extract and purify your HB vaccines from these antigens. So commercial and pharmaceutical products, um, like insulin, which controls glucose in humans, by recombinant DNA done cloning of human insulin gene put into E. coli. Availability of insulin devoid of getting byproducts by animal slaughtering. So we can do work at the animal level and get the results done easily. Human growth hormone, HGH hormone, are being produced with the help of this technique with 121 amino acids, 2 to 115 Dalton molecular weight, role in growth, regeneration, and differentiation. Examples is dwarfism, treating by injecting these. Uh, so dwarfism could be treated by injecting these genes. So here you can see there is sequence of 24 amino acids of HGH. It's being produced, and there is sequence of other sequence on sidewise, and we introduce and we cleave it with the E. coli cleavage, and then inserted your concerned genes of 24 amino acids into the vector genome, and then into the vector they are inserted into again into E. coli to further production of it. So production of recombinant growth hormone is done like this, HGH hormone. Then there's interferons. Interferons are the group of proteins that interfere with the viral multiplication of viral multiplications or replication. So by recombinant DNA, they are capable of making interferons, alpha component of which have role in curing lymphoma and malignous leukemia also. So antibiotic production is also available in this case. Artificial prepared antibiotics are also available. They denature harmful living pathogens. The first antibiotic that was produced in 1928, penicillin, in that case. So they are monoclonal antibodies which are produced by hybridomer technology. So they are the antibodies which are which specific proteins produced by the immune system in response to presence of specific antigens. Monoclonal antibodies produced from single clone of antigens. That's why they are monospecific in nature. And production through hybridoma technology. Application is they are used for diagnosis of disease, pregnancy, and treatment of cancer. So there are antigens which are being produced in the immunizations, and we isolate these immune cells, and then antibody forming cells are produced, and then we fuse them with the tumor cells, and these tumor cells are known as now hybridomas, and we screen for the desired antibodies because we can have large production of antibody because from these tumor cells. And then antibody producing hybridomas are cloned, clonal expansion is done, then you have a monoclonal antibodies being produced at the end. There are a lot of diagnostic kits are being produced with the help of in this recombinant DNA technology. So infectious diseases diagnosis mainly depends upon isolations and identifications of pathogens, which may take several days. So development of diagnostic kits to identify pathogenic problems by knowing their organism specific DNA has provided rapid, specific, correct diagnosis. Like recently, we have also for COVID-19, and so far their diagnostic kits are available for AIDS, cancer, foot, mouth diseases, tuberculosis, etc. So you add your specimen into the hole like this here, then wait for 5 minutes. So you will have either positive or either negative results, or sometimes you have invalid results. So based on ELISA and PCR principles, for example, in HIV patients, diagnosis conducted by antibody test uses recombinant HIV proteins to measure antibodies in the body that proliferate when there is HIV infections. Then DNA test uses reverse transcription polymerase chain reactions (RT-PCR) to detect presence of HIV genetic material, and this technique was developed using recombinant DNA of molecules and analyzing the genome sequences. More is DNA fingerprinting is also possible. That was developed by Dr. Alec Jeffries, because every person has different unique pattern of DNA, and based on that we can identify the unknown particles on the crime scenes, or for the child percentage establishment, for the detection of racial group. So fingerprints are detected on the basis of number of high polymorphic genes, that is VNTRs. So first you take your blood sample, then DNA is extracted, DNA is cut using resection enzymes, then DNA fragments are separated bands using electrophoresis gel, then DNA bands are transferred to the nylon membrane as a technique known as southern blotting. Then further DNA binds probes into a specific DNA sequence on the membrane. First radioactive DNA probe is prepared, then excess DNA probe is washed off from the nylon membrane. So at this stage, radioactive probe is bound to the DNA pattern on the membrane. So X-ray film is placed next to the membrane to detect the radioactive pattern. So you can have X-ray patterns like this. So we did this in southern blotting in detailed manner. So how we do from step 1 to step 11 in details that day. So then gene therapy, in which you inject functional genes into cell to replace missing or defective genes in order to correct genetic disorder. So a gene that is inserted directly into the cell usually do not function. Instead, a carrier called a vector is genetically engineered to deliver the gene. Gene therapy may be in vivo or ex vivo. There are some healthy risks. Toxicity, inflammation, and cancer could be possible because of gene therapy. So there is a direct delivery and cell-based delivery. In direct delivery, your therapeutic transgene is packaged into the delivery vehicle such as viruses, and they're injected into the patients like this. Or you can have a cell delivery based therapeutic transgene uh, is first being therapeutic transgene is packed into delivery vehicles such as virus, And then the therapeutic gene is introduced in delivery cells such as stem cells, often derived from the patient cells. So your virus cells and stem cells from the patient, they are mixed together and they multiplied in the laboratory, and then they re-administrated in the patients. So there are two ways to do so. So that was about gene therapy. Any questions, students, so far? Ah, so you're all talking about free journals at the moment. Let's concentrate on the studies first, then we discuss about that also later. Don't worry, don't worry. So we have discussed everything from the health perspective. Now comes the environment perspective. So we can use recombinant DNA technology environment to clean up the environment, to remove the hazardous compounds present in the environment. So by recombinant DNA technology, we can remediate environmental solutions, pollutants for like some harmful chemicals that are not diagnosed, that are not digested and degraded. They are partially degradable, like TNT chemicals. In this, nitrogen is reacted with oxygen, and superoxide is formed that is toxic. So knockout of the monodihydroascorbate reductase gene that will create resistance against TNT. So you add that gene, knock out the monodihydroascorbate reductase gene, which will create resistance against TNT in our solutions, in our pollutant. And you can do arsenic particles are toxics and they are present in the soil and that is harmful for plants. PVACR3 gene is inserted into Arabidopsis, and this gene is expressed will give rise to tolerant plant to the arsenic. So in Arabidopsis thaliana, phytochelatins resist the movement of arsenic in root cells and phloem cells, and reduxin is present in them which reduce the arsenic also. So all these reasons are reason uh, to reduce to get rid of the arsenic and to get protected from it. Then waste product of agriculture have cellulose that do not easily decompose. By using recombinant technology, we can convert cellulose into sugar by breaking down of cellulose. So for that we need cellulose enzyme. We add that into the vector, then incorporate into the bacteria. So expression of this enzyme will enhance the degradation of the waste that contains cellulose with the help of your cellulase enzyme. We can also degrade plastic by enhancing the genetically modified organisms, GMOs. We can also degrade oil spills, organic waste by a modified strain of Pseudomonas putida, able to degrade chemicals in oil spills. We can also use them as a bioindicators. So, for example, bacteria like bioluminescents that give off light in response to several chemical pollutants. So, this is also possible, one of the trait of the organisms that could be used to degrade the pollutants. So these are being used to measure the presence of some hazardous chemicals in environment. Other genetic sensors can be used to various chemicals, contaminants undergoing trials, and include sensors that can be used to track how pollutants are naturally degrading in groundwater. For example, mercury resistance gene (MER) or toluene degradation (TOL) gene is linked to the genes that code for bioluminescence within the living bacterial cells. The biosensor cells can signal extremely low level of inorganic mercury or toluene that are present in the contaminated waters and soils. They are emitting visible light, measure the fiber optic fluorometers at the end. So that was something about environment. Now comes the applications of recombinant DNA technology in agriculture. So they can be used for insertion of genes in plants, not only from the related plant species, but also from the unrelated species such as microorganisms. Users used for the production of transgenic plants with higher yield uh, for nutritional values, increased resistance to stress and pest also. Improve yield, stress tolerant plant transgenic plants such as biopharmaceuticals are also we are able to produce with the help of these plants. So plants with improved yield, genes are inserted into the plants to increase their yield. At the Japan Institute, agrobiological resources added maize photosynthesis genes to rice, so which has increased the yield to the 30 percentage. And Mexican scientists have genetically modified plants to secrete citric acid from their roots in response to the slight increase in acidity soil particles and released and made available to the plant in that case. Stress tolerant plants, plant resistance to environmental stress have been produced using a recombinant DNA technology to tolerate the abiotic stress. So, tomato, canola plants that can tolerate salt levels up to 300 percentage greater than the normal. So, here on the left is the tolerant plants, and here is the susceptible plants. So, we can grow tolerate salinity, the salt tolerance by 300 percentage. Herbicide resistance plants are also possible. Uh, Roundup is an herbicide, it kills almost all species, but using recombinant DNA technology, using EPSP synthesis and gene that have been introduced to crop plants and into like cotton and soybean, you can save your species for the longer run. Insect resistance plants like cryogens, BT cotton, that be very famous, produced with the help of bacteria Bacillus thuringiensis. We isolate from that bacteria and insert into our species. The plant is modified using this gene. So cotton, rice, maize, potato, brinjal, cauliflower, cabbage, they all have been produced using the BT genes have been developed so far. Then disease resistance plants are modified to produce resistance against diseases. Example, tobacco was first modified to produce resistance against tobacco mosaic virus. So on the left is the tobacco mosaic virus plant, and right hand side is the wild type one. So this is the resistance left, right is the wild type. So production of biopharmaceuticals is also possible with the help of plants. Recombinant DNA technology used to produce a plants that will generate a seed that express a desired therapeutic protein. Then seed stock is used for producing the desired proteins. The desired proteins can be extracted from the seeds. Example, in corn biopharmaceuticals are produced. Edible vaccines, the genes encoding antigenic proteins can be isolated from the pathogens expressed in plants. Producing antigens can be eaten for vaccinations, immunization. In banana and tomato, edible vaccines are produced. Production of secondary metabolites. Arabidopsis plant is modified. Polyhydroxybutyrate is released by Arabidopsis. PHB is a biodegradable plastic. So applications, this was done in the case of plants in agriculture. Now comes the last, not the least, applications of recombinant DNA technology in industry. How recombinant DNA technology is helping in the industrial side. So traditionally, industrial microbiology is merged with the molecular biology to yield recombinant process for the industrial production, so such as primary and secondary metabolites, protein biopharmaceuticals, industrial enzymes are formed for the industrial usage. So important primary metabolites like amino acids, nucleotides, vitamins, solvents, organic acids, their production in energy production and substrate utilizations, which are essential for the growth. These are for the primary metabolites. So vitamin B12 is produced commercially by direct fermentations using the fungus Ashbia gossapi, and it is also utilizing Streptomyces species such as Streptomyces griseus for the production of vitamin B12. So vitamin C is produced by using gluconobacter oxidants. P carotene is produced by the members of Coniferaceae family of Phycomycetes. Then amino acids, E. coli and cloning vector PBR322 were used to increase the genes for the production of amino acids like glutamic acid, lysine, phenylalanine, valine, and L-glutamate, MSGs, and L-lysines, mostly used as a feed and food additives. Then this is the golden rice technology has been explained here, in which the genes that give golden rice's ability to make beta carotene in its endosperm come from the Daffodilus and bacterium called Erwinia iridoviria. So you have bacteria and Daffodilus. So you mix the genes together, and these genes, along with the promoters, are inserted into plasmids that occur inside a species of bacterium known as Ectobacterium tumefaciens, and these agrobacteria are then added to the petri dish containing rice embryos. As they infect the embryos, they also transfer the genes that encode the instructions for making beta carotene. The transgenic rice plants must now be crossed with strains of rice that are grown locally and are suited to particular regions of class, climate, and growing conditions. So they are locally important varieties in this case. So enzymes are biologically molecules that catalyze the increase the rate of chemical reactions. Recombinant enzymes playing vital role in food industry. Amylase and proteases are one of the example which are producing in fungi and plants high fructose corn syrup, and biscuit manufacturers are using them proteases for the lowering the protein level of flours. Then this is for the amylase production, showing you steps involved in the efficient amylase gene, that insertion of the genes into appropriate vector system. So your promoter, you have double-stranded plasmid DNA expression vector. You cut it with restriction endonucleases. An amylase coding DNA sequence is inserted, then introduced into recombinant DNA into cells, then leading to the overexpressed mRNA and overexpressed protein amylase. So this is how we do in the bacterial system to produce your amylase production with the help of recombinant DNA technology. So you can see these are the all things that we have discussed so far since we start the class. So it's all about how recombinant DNA technology is playing a role and has such a great impact, whether it's agriculture or whether it's a health system or it's a plant system or industrial system or environment. So it's being used in every factor quite indispensably and quite what do you say, have a high impact in our day-to-day life also. Then comes the chymosin. These are derived from the rennet used to manufacture of the cheese. So amino acids, peptides are used for the fermentations, and proteases split the polysaccharides and proteins into the malt. Prochymosin genes are isolated from the young calves and they are transferred into the plasmids and introduce microorganisms. And on expressions, prochymosin is activated into chymosins, producing 100% pure chymosin in that case. So first step is the milk clotting process in cheese making, in which K canalitic enzymes contribute to the micelle precipitations because of specific towards the K casein for its purpose. So here you can see cheese production in recombinant chymosin. So you have a gene for chymosin isolated from the cow. So you isolate the gene from there. Then you have a plasmid which have rejection enzyme sites, and then you combine these together. Then you join them together with the help of ligase, and you put recombinant DNA into the bacteria, and then transformation is happened. Then chymosin protein is expressed in bacterial cells. Isolate chymosin protein. Then from there you can have isolated chymosin used to make cheese further. Further, biofuel industry, cellulose used to break down cellulose into sugars that can be fermented. In molecular biology, section enzymes, DNA ligase, polymerases used to manipulate DNA in genetic engineering, important in the pharmacology, agriculture, and medicines. Also used in the forensic science. So that's it for today class. So now it's question answers time. The ethylene gases which is released by plants on a GM, will it affect is in a good way or a bad way? So I mean, ethylene so far it's a hormone, yes, which helps in the uh, ripening of our fruits to be done faster. So what if we have ethylene hormone itself in the plant overexpressed, so they will ripe faster. So farmer doesn't have to wait longer to get the fruits ripe, and they can go for it, go for the selling. So that is also one thing is possible. So student, today was your last day of your present of your day, 10th day, because we have one day a Sunday as off. That's why it's become from 16 to 17. So you can contact me anytime. I will try my best to answer your questions. Sometimes I can, sometimes I can't. Please text me. I can answer on your whenever I am free. I will try to answer your things on the text. Yeah, so I will share today's presentation. So Rubisco enzyme. Have anyone done that before? Rubisco enzyme. I mean, any enzyme is possible. You just have to insert it into the plasmid and then into the E. coli bacteria, and it's good to go then. So yeah, somebody did that research in 1991. Quite old. Expression of endogenous foreign ribulose biophosphate genes in Rubicus deletions mutant of rhodovector. This is a bit complex. Cloning is always complex to make it done at the end, to see the which vector will fit and how you do it. So this is also has been done in research in earlier time, 1991. Yeah, by Falcon and Robert Tabita, I think from Ohio. Are you working with the Rubisco enzyme in particular? Okay. No, no, you can't. Same paper not possible in different journals. It has to be published one paper, one thing has to publish in one journal. That will come with. That's a good idea. That has to be available for one journal, and you will have one thing. So yeah, I mean Ribisco, like how the Biocon company came out. So Kiran Majumdar ma'am, what she did, she produced papan enzyme in her garage area. So she produced that as a commercial. So their company was a spin out from these enzymes thing also. So you can try, why not? So the enzymes has many big functions. So many companies are producing them, and you can initiate your own wealth sector with that. I'm sorry. God bless me. The air is changing with this cold weather. Please take care of self. Um, Processing of publishing a paper. So, बच्चे first. Oh, the biochemistry of Rubisco. बच्चे, I'm not into the Rubisco's biochemistry. You have to like, I can help you. Like, you can check, right, biochemistry, Rubisco's biochemistry. Then from there, structural mechanisms of Rubisco activation by carboxylase oxygenase. Carbamylation, you can activate the lysine activation of Rubisco by carbamylation. So you can go through this general, you can get the answer. So, but in my time when I published, it was very tedious method. So I published my paper in Nature's Scientific Reports. So first I have to write my paper, of course, with the methods. I have to create different figures, uh, some different figures. Then figures has to be as per the review, as per the general prescription, how the figure should be, how the legend should look like, and how many figures are allowed. So there are many. There were many. It took me almost 1 year to publish this paper, because once you publish, your material methods, your references, acknowledgement, uh, things get. Then you get three reviewers. Uh, you get three reviewers who give you back their reviews about your journal, about your work, about your paper. So then you have to work on the things. If they ask you to do some experiments, you work on experiments. Then afterwards, you go for more experiments, then more results. Then you send them and then ask for them if everything is fine. Then at the end, your paper is accepted, and then it's ready to publish. So mainly it depends upon your boss, your PI, your principal investigator, who is dealing with that with your research. How good they are good with the general people, and how good their connects connections. and based on that, things are possible. Yeah, it's mostly internships are for the MSc students because they are more mature and they can do handle the lab things more better. For the bachelors, it's difficult. How to draw biological figure? Biological figures, how to draw? On PowerPoint. I use PowerPoint. Everything, all my research, everything I did was on PowerPoint. And now all these figures that I have created in my paper, I use PowerPoint. That's it. So these tables, I use software called Prism. The software for creating scientific figures. Use software, you can use Prism, which helps to create things automatically for you, and it's very easy and fast. So basically, I use Prism, PowerPoint, WordPad. So I didn't use very hi-fi softwares for my PhD. So to for Western blotting, I use Images software. So that's it. Is it necessary to cite every fact given in the reference books in the review? No, it's not. Mainly, it's a story. Review is like a story where you are extracting all the information from all the journals and you bringing them together in your review. So it's like a story that you are telling. So just create a story and then give the references from the people who have taken the things. Then keep writing, keep writing. Space biotechnology. Wow. I think there's no courses available at the moment, but it's interesting. It's very interesting. Ah, there is master's degree in biotechnology, space medicine. Wow, I am amazed. So, which university is this? Suffolk. No, 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 not this one. Regenerative medicine, biochemical engineering, biomedical engineering. Mainly three courses I could find. So, this is a website, findmasters.com. If you're looking for some courses, go through this link. But citations means when you cite papers. Um, so somebody wrote this journal. So they wrote 7, 8, 5, 3 here. So they cited some journals. MSN has been used as an ideal carrier for various biogenic agents. So this is journal number 3, reference number 3. So 1, 2, 3. So this is being carried out from here. So this is how they cite the paper. So they took it from this paper and cited like that. Yeah, NASA also provide that. That's true. So somebody send me. So students, if you have no further questions, shall we end the course today? Or we can still have extend our Q&A more. No, it's not. Hyperlink is not necessary. So it's depend upon which generals you are going. So according to that, you have to write. Somebody wants, somebody don't want, but mostly it's not required. Hyperlink is not required. In my case, in my general, when I did, it was required. So we have to have DOI link, and so that was a style that this general required. So that person could go to three these papers. Bache, Novindo, I got your email also. I couldn't reply. Sorry for that. You have to submit on your own actually. So we will not publish it. So you have to do it on your own. So just go to the free generals and try to ask the editor if they can accept it. Then if it is accepted, then it's good to go publish. So our task in the internship was to to get about have an idea of about writing review while we're doing the various techniques in MBBT and their applications, our day-to-day life. So that was our main aim to go for. Statement of purpose, SOPs. Mainly SOPs are the statements in which you have to be genuinely honest that why you want to be in that course and what will be the benefit that you will get from the module of that course. So go through the modules, check the courses that they're providing, and write about your motivation, genuine motivation, why that course will be really beneficial for you. Only then things will be something concurrent and definite for you. Some path will open only then. So take care while doing so though. So internships letter will be provided and certificates will be provided instantly, maybe by 25th of October. Internship letter when you submit your review letter with the topic of it, only then internship letter will be given to you back. Yes, I mean no, it is not required to have single author. There could be three or four authors also, so that will be no problem. It's up to you how you want to do it. So for your internship letter and certificates, please ask Vatsal sir and Shwani ma'am. They will be dealing with that. No books or websites in writing review. No books or journals. So it has to be from the review. So from the journals itself. Just email us the paper, or they will ask to the Edufabrica website. Ask Vatsal sir how they should be done. So they will be a better person to talk about that. Yeah. And please, before you leave, there are around 56 students. So subscribe to the channel This will really help me to grow more on YouTube and to bring more things for you, good things. So I was thinking of creating some unique videos, so of these techniques that you have learned, maybe in the small one pattern, so that you can directly look at the techniques and go for it. So I'm trying my best to do so. Let's see. Just try to send us both. No worries. Thank you very much. Thank you very much. Thank you. Thank you. Thank you, everyone. So, okay, I will do it. I will do it. Thank you for suggestions. Thank you, everyone. Thank you. So, Simran Jan, you have a question in my email. I'm a student of MBBT group. I'm interested in doing PhD from Germany. So, I wanted to know what is the procedure of applying scholarship. So, we can apply for. How can we upgrade our CV? Which type of scholarship? There are so many questions. It's hard to reply you via email. I'm currently in the MSc second year of my biotechnology. So if I drop a year, then what activity should I enhance my resume? Please guide me. I would thank you. I would suggest to apply to the professors directly for your PhD, and then look for the answers how they reply in the field of your master thesis you did in. So you will definitely get some answers from the professors. By the time you apply around 10 or 20 applications, you will get to know where you are standing. Professors also reply to you back in very nice manner. Yeah, email is fine. Sometimes I can't reply. So also text me. You can write me on WhatsApp. So WhatsApp is better. So I can reply instantly if I'm free. But most of the time, it's hard to reply. So sorry for that. But I will try my best. So don't drop your ear. I would suggest you, and go for industry. If nothing is possible, go for industry. I will always suggest go for industry. Industry is the best thing to go for. Okay, students, then sayonara, konnichiwa is to say welcome actually. Bye bye. See you again somewhere in some workshops. It was pleasure seeing you all. Yeah, yeah. Postdoc is necessary. Yeah, it's hello. Konnichiwa. So, Dankeschön, Namaste, take care, stay safe, stay healthy, go for regular exercise, eat healthy, and have a mental balance, good mental balance to see what is clear for you. Do meditation, wake up early in the morning before sunrise. Yeah, take bath, be clean. So take care of self and the near and dear ones, everyone, so whosoever you contact with. Be the light, spread the light. Thank you. Bye bye.